Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about antibody structure. So first you have to know that what is the antibody. So antibody actually is a protein or polypeptide that made by the plasma cells in response to an antigen to neutralize the antigen. So this antigen might be the bacteria, virus, or any kind of non-functioning protein in our body. So let me draw a picture here. So we know there are a lot of immune cells. So there are some special kind of immune cells, that is the B cells. So B cells, it has some surface receptor. This surface receptor is the B cell receptor. So this is the surface receptor. Let's assume that. And also there are special kind of cells, that is the antigen presenting cells. And these antigen presenting cells, their job is to very specific to represent the antigen. So it represents the antigen by the MHC molecule. So this is the MHC molecule, and by this MHC molecule it represents the antigen, and this antigen it recognized by the B cell surface receptor. Alright, and then B cell then with other co-stimulatory pathways, B cells get activated, and then B cells, the couple of B cells that transformed to the plasma cells. Plasma cells is nothing, is the same kind of B cells, but it has some structural modification. And these kind of plasma cells, they are very much able to generate special kind of antibody against some specific kind of antigen. So these plasma cells, it produce antibody, and this antibody is very much specific with this kind of antigen, but not any other kind of antigen. So it can bind only this special kind of antigen. Alright, so this is the basic principle about this kind of antibodies generation. So let me show you the structure of the antibody. So next is the antibody structure. So here, as you see in the green color part, these all are representing the constant part. So in the constant part there are different kind of domain. So here is the heavy chain. So in the heavy chain there are three different domain: CH1, CH2 and CH3 in the constant part. So CH means heavy chain constant part, and also there are the heavy chain variable part, that is the change always. So VH, it represents the heavy chain variable part. So this part is the heavy chain constant part, HC, heavy chain constant part, and this part is the heavy chain variable part. Alright, so this is totally the heavy chain, and just beside the heavy chain there is also a small chain that is known as the HCl, or the light chain constant part, and here also the small chain, that is the light chain variable part. Alright, so in the green color also in the constant part. So as the name suggests, this constant part always same, but according to the antigen structure, this variable part always changes according to the different kind of antibody. And also in the variable part there are some special kind of sequence. That region is known as the CDR1, complementary determining region 1, 2 and 3. And these are very much important sequence that change according to the antigen structure. So for special kind of some different kind of antibody, this structure CDR1, CDR2 and CDR3, these are change always depend on the antigen structure. So here also there is the hinge region. So this hinge region is just to hold the FC and FAB region, and this hinge region are made by disulfide bond. And this part also is known as the FC part, that is the constant, that is the fragment constant part, and this part is known as the FAB, or fragment antibody binding part. Alright, so next is the antigen. So this antigen, this special kind of antigen that made this kind of special kind of antibody against this antigen. So this antibody is very much specific to this kind of specific antigen, but not any other kind of antigen. But if you see this antibody. So this is type A antibody and this is type B antibody. So this type B antibody, these are pretty much different with this antibody, because you see here the antigen structure in both cases is not similar, it's different, right? So because of this antigen structure, this antibody is very much specific with this kind of antigen, against this kind of antigen. But this antibody, it does not work against this antigen. And also the same, this antibody, it does not work against this kind of antigen. All right. So next is the one of the example I'd like to compare. So so this is the fruit picking stick. So as you see, this this guy is picking the fruit by this stick, and this stick, if we compare with this stick, so this stick actually it has the constant part, right? So if we compare with this antibody, so this is the constant part. So this is just to hold this part, and this part we can change it according to our needs, right? So sometimes also there are different kind of fruit picking stick, but this part always same. So this part just to hold this part and to stable and to make it stable. And also if you see the another kind of different kind of spoon, also here this kind of spoon, it has some constant part. So this constant part always same in all kind of spoons, but here there is this head is always change on depend on different kind of situation, right? So this is, if we compare with the antibody, so this antibody also it has the constant part, and this constant part is just to hold this variable part, and this variable part it change according to the antigen structure. All right. So I hope that this example it is very much useful. And next is the epitope and paratope. So what is epitope actually? So epitope actually, this is the special kind of sequence of the antigen. In that sequence the antibody gonna bind. So in this antigen A and B, this is the epitope. So epitope is the special kind of sequence, and this sequence the special kind of antibody that gonna bind here. And also for, you see there are 4 different kind of antigen, but epitope structure is different in all cases. And also the sequence of this epitope is changed, it's not the same, is always the amino acid sequence it's changed. And for this special kind of antigen, for each against this each specific kind of antigen, there are each specific kind of antibody. So here in all 4 cases there are 4 different kind of antibody. So here, so if you like, if we say that this antibody it cannot work with this antigen or this antigen or this antigen, this antigen is always specific against only this kind of antigen. All right. And also in the antibody where the epitope bind, that part is known as the paratope. So this part is known as the paratope. So see, this is the epitope binding part, and all cases this epitope binding part there is some specific region, and this region known as the paratope of the antibody. So this is all about the antibody structure. So I hope this video will be helpful, and if you like this video, kindly hit the like button, share it, and if you have any queries, kindly write in the comment section. And please, if you like this video, subscribe my channel. Thanks.